ICAR ARS NET exam and ARS prelim exam, they were conducted together. So this exam is pretty much easy to crack when compared with CSIR NET examination, because the syllabus include only the basic topics that we already studied in your degree and masters. So the one of the important feature of this exam is that the ICAR board conduct this exam in nearly 55 subject, nearly 55 discipline. So it is very easy to crack the examination in one particular discipline because it include only the topics that you have already studied. Just one refreshment is required. Today, I am going to talk in detail, little bit more about the agricultural microbiology discipline because I belongs to it. It is very easy because the syllabus include hardly one page, and you can qualify the exam very easily if you are adopting a preparation strategy in such a way that. So I will discuss what and all the important topic that you have to focus in each module and how your preparation strategy should be. The syllabus of agricultural microbiology, the entire syllabus is divided into 5 modules. So you can see here hardly one page and it includes 5 modules. So always start your preparation with first module, which discuss the history of microbial world. So when you are starting your preparation with history, in almost all the microbiology textbook, you'll have a detailed history of microbiology. But if you want a little bit more details, of course, you can refer Prescott Microbiology textbook or Brock Biology of Microorganisms. Both those are advanced textbook that I will refer to prepare for your ICAR NET Agricultural Microbiology exam. So in history, you study in detail about the contributions of different scientists, Louis Pasteur, Coast, Robert Anthony van Leeuwenhoek, and theory of spontaneous generation, how it is disproved, germ theory of disease. So step by step, year-wise, you study all the important parts of our microbiology history. Don't forget the year. So every year you should mark or highlighted here, because almost all the questions in ICAR NET exam is one word questions and multiple choice type. So they may be asking some term, some scientist name, some year, some microorganism name, like that. So don't require so much calculations, or it won't require so much analysis and all. You can, if you know the term, if you know the scientist name, if you know the year very well, if you are by hearted it very well, you can answer it quickly. So then you should focus more on the theory of spontaneous generation that I already told you. Next comes the different types of prokaryotes. Here given a huge list of prokaryotes. So all those prokaryotes characters, their habitat, don't forget to study the example for each group. So that you have to do. Then you have to focus more among this group, nitrobacteria and nitrogen fixing bacteria. Anyway, we are going to study this nitrogen fixation and nitrogen cycle topic in some other module, but keeping it in mind that this nitrogen fixation, nitrogen cycle and nitrogen fixing bacteria, they are very relevant in ICAR ARS NET examination. So you study the general features of all this group of microorganism. And one important group like nitrogen fixing bacteria here is methanogen. In you can expect at least one questions from methanogens group, so you should focus more on it. Similar to that thermophilic group also. Then another important thing is that the identification, what are the different identification strategy that we can use, or technique that we can use for classifying the bacteria, no, our microscopy, cultural characteristics, staining reactions, biochemical reactions, some molecular typing. If any no, recent advancement is there in that field, you can refer and you can include that also in your preparation. Then the important part in the second module is the interactions, microbial interactions. You start your preparation with microbial interaction in second module. It includes symbiosis, synergism, commensalism, parasitism, amensalism, antagonism, and predation, almost all the interactions, and give examples for these interactions, and all the characters of these interactions and their significance in maintaining the microbial population in our environment. That all has to be studied. Then microbial metabolism, like aerobic and anaerobic respiration, electron transport chain, microbial photosynthesis, and oxidative and substrate level phosphorylation. So this is the important part that you have to focus in unit 2. Then unit 3 is soil microbiology. So in unit 3, there are a lots of general topics like what is rhizosphere, soil health, root exudate and its components and all that you can read and move. And you have to focus more, or you start your preparation with nitrogen cycle. Each and every steps of nitrogen cycle, especially biological nitrogen fixation, its overall reaction, enzyme participating and microorganism participating in the reaction. So everything require an advanced reference. So you always start your preparation in nitrogen cycle topic with a textbook like Brock Biology of Microorganism. Don't stop with the basic things what you have already studied. Just refreshment is not sufficient, because when I am writing my exam, there were 3 questions from this nitrogen cycle and nitrogen fixation area. So you can expect minimum of 2 or 3 questions from this area. Focus more, prepare well, and you should thorough with the concept, of course. Then biofertilizers, different types of biofertilizers, different group of microorganisms that we can use as biofertilizer, significance of this biofertilizer, some commercially available biofertilizer, their commercial name, their preparation, their formulation. So all those things you can study. Similar to that, bioremediation is another concept. Then you give some example of plant growth promoting rhizobacteria. So you list some microorganisms, they are commonly present in the rhizosphere region, and they act like a PGPR, plant growth promoting rhizobacteria. That is also very much important in this module. Then unit 4. In unit 4, microscopy. At least 1 or 2 questions you can expect from this area. In microscopy, different types of microscopes you have to focus, and why we need to use different types of microorganisms, their purpose of using, how it works, their principles, and you just draw the structure of every microscope briefly, and its components, its purpose, everything has to be mentioned properly in your notes. So in my examination, there were questions related to dark field microscope, its purpose. So you can prepare in such a way that everything from structure to function, everything is included in this microscopic topic. And microbial assay of vitamins, enzyme, and antibiotics that you can study. Then sewage water treatment. It is very important, sewage water and effluent treatment. So that we are studying from our basic degree classes onwards. So you can just refresh the topic, different steps involved, different group of microorganisms involved. So that so on. Then waterborne diseases. This topic, the covering the entire group of waterborne diseases, their treatment, their spread, So it will be very huge for you. So study some basic general waterborne diseases that commonly seen, their causative organisms, their epidemiology, their symptoms, and all you can cover, like cholera disease. Okay, then unit 5. I would like to say that this unit 5, it was a little bit difficult for me to prepare. So I given more time for this module because it is more about the industrial microbiology, like fermentation process, different types, batch, fed batch, and continuous type fermentation process, different types of downstream processing. Simply in syllabus, it is given downstream processing, but we know that different levels, different types of downstream processing it is there, so that you have to study. Then you have to study about single cell proteins, different groups of microorganisms that can be used as single cell proteins, some probiotics, and some microorganism produces hormones, and biofertilizer topic that we already covered in previous module. And you focus a little bit about the biopesticides also, like bioremediation. Along with that, you can study phytoremediation, a simple topic that you can cover. A brief outline is required related to this phytoremediation and how it works, and that and all you can study. Then the next part of this module 5, it mainly include food microbiology topics, like some preparation of some fermented food like vinegar, wine, sauerkraut, pickle, cheese, yogurt, etc., how we can prepare that. So if you are referring a Fraser, if you already studied your Fraser for in food microbiology textbook by Fraser, so that textbook is much more enough to cover all this part. So you study some key microorganisms that are used for the preparation of this fermented food, and how we can preserve food, like use of high temperature, low temperature, asepsis, canning, and loads of methods are there. Everything chapter wise it is discussed in that textbook, Fraser textbook for microbiology, that you can refer. Then some microorganism, they mainly causes contamination and spoilage of food. Then you briefly outline the foodborne illness, as like said, like our waterborne disease, foodborne disease, only some key diseases like botulism diseases that we have to focus, and don't study the entire foodborne diseases. It will waste your time. And the remaining portion in this module that discuss the microflora of meat, fish, egg, fruits, vegetables. Actually, I leave this part, but it is optional. It is left to you if you want to study this portions, because we know that the microflora of meat means there are different groups of microorganisms. It is very difficult to remember the names of all the groups separately in case of different types of food. So if you have that memory, and if you want to cover the entire syllabus, and then you can obviously can prepare well for this also. The same textbook by Fraser you can refer. But otherwise, leaving one or topic, if you are very much thorough about the all other topics in or almost coming in all the modules, then don't worry, leaving one or two topics don't make much changes in your marks. So that also you can do, that left to you. Okay, I will tell you how to prepare your notes for ICAR ARS NET exam. And first of all, I would like to say one thing. Don't prepare a note that is too much. Like an essay, you don't need to prepare note, because you cannot revise the entire topic just 2 or 3 days before your examination. So in order to remember the concept and only the required portions or required terms very efficiently, you can make your note like a question answer type. See here. So if you're studying peptidoglycan. Don't write the entire history about peptidoglycan. You know the basic structure of peptidoglycan, so no need to draw the structure of peptidoglycan. If there is some terms that you don't know, then make a question out of it and write its answer below. Then if you know, for example, where peptidoglycan is synthesized, that you already know, if you know that, don't waste your time writing that questions. Instead, if you don't know, then make a question out of it, write it below the answer. Similarly, how the peptidoglycan is transported to the extracellular site, which protein is involved. If you don't know that, immediately write that question and write the answer below. So instead of writing the entire essay what you read in your textbook, what you understood, so it won't works for a competitive examination. So this is an easy way of making the questions. Again, we can short, we can shortlist, and we can select what are the important questions that I don't know, its answer I don't know, or only those questions you can write in your notes. And whenever you want to revise, you just quickly revise all these questions, and automatically hide the answer, and automatically you will get to know what is its answer. You can like preparing for PSC examination, it will work. So at the end of your preparation, you have a small book with a lots of questions from all the required topics given in the syllabus. So that is enough to qualify an ARS preliminary exam as well as ICAR ARS NET exam. You can score a very good mark if you are following this technique. And I am assuring this because I followed this method, and I tracked my CSER. I tracked my ICAR ARS NET exam with this preparation strategy with the first try itself. So just try like this. You minimize your time, minimize your effort for writing the notes, but make a note within a minimum period of time. But it is very much essential, since the exam pattern is very simple, one word type, multiple choice questions, and don't much analysis required. So it's such a way, this is the apt strategy for studying the ICAR NET exam.